This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 15 for January 12th, 2009. I'm Vincent Racaniello.
And I'm Dick Despommier.
And I'm Alan Dove.
How about that? 3 people today, and Dick is back. Welcome to This Week in Virology, the podcast about all things viruses. And welcome back, Dick.
Thanks, Vince.
Been in India, I understand.
I have. Spent the last month in India, as a matter of fact.
Did you like it?
No, I loved it. I actually had a great time. It was fantastic. It was a three-ring circus from start to finish. Trip started out in southern India, in Bangalore. We took a little side trip down to Mysore, came back up to Bangalore, and then went up to Pune. And then from Pune, we went to Aurangabad, and then we went up to Khajuraho, and then finally to Agra. Of course, you have to go to Agra to see the Taj Mahal. And then we went to Delhi and stayed 4 days in Delhi and had some fantastic cuisine, no matter where we went, no matter how expensive or how reasonable the restaurant, even restaurants that you might not want to consider going into based on the fact that the bull just walked out of the restaurant. The one thing I was quite amazed at was that, when they tell you that everyone worships the afterlife over there to the extent that they believe that all those cattle are ancestors, people who have lived other lives before that, those people actually believe it. And I don't question that at all, by the way. I'm not actually saying anything derogatory here. I was just amazed at seeing how many cattle there actually were in India. Just incredible. And they have the right of way. So if they're in the middle of the road, all the cars either stop or they go around them. The cattle are so used to their traffic that they don't move. These were goats, sheep, cows, elephants. We saw everything, and my eyes are still wide open. I'm absolutely amazed at what I've seen. It reminds me of a line from a John Lennon song that says, "The more you travel, the less you know." And it's absolutely true. The more I have seen of that part of the world, the less of my own world I understand. And it should be that way because, you get insular. And the biggest thing in virology that I saw while I was over there, Vincent, you'll be happy to know that I did pay attention at least to a little bit of science while I was there.
What did we get?
What we got was a public health program with regards to vaccination for polio. And what they do is they actually print along the side of the building with paint the date that they visited that house. They list the strain of the virus that they gave on the sugar cube to the kids, and they list how many kids they gave it to.
Fabulous.
And those paint marks stay on that building for a long time. So I took a lot of pictures of those because I thought you'd be interested in them.
Definitely.
And you can use them in your teaching and put them on the blog and show the people.
Virological graffiti with a purpose.
And you got it right. The two things that reach all the people are polio vaccine and Coca-Cola.
That's right. And so the marking of the house is to show where it's been so they don't go back and ask and waste time.
Correct.
Right?
That is exactly right. Now, they used to do that for malaria, by the way. When they sprayed with residual spray with DDT, they used to mark them the same way for that as well.
Well, what did you actually go there to do? Give lectures?
I did. I went to give a lecture to the Indian Institute for Architecture. Actually invited me to give a talk about vertical farming. So I was honored to be asked to go. Had a great time. Met a lot of interesting people.
Are you? Is someone there thinking about building a prototype?
They are, in fact.
So you might go back.
I asked them what the biggest agricultural problem was facing India, and they all had the same answer. They said the monsoons, and I said, "But you still get the monsoons?" And they said, "Yes. We used to get monsoons for 3 months, a gentle rain. Now we get the same amount of rain in 2 months." That's a huge problem.
Why has it changed? Do we know?
They think it's changing because of all this greenhouse gas that's been put into the atmosphere. So rapid climate change has had its impact on Indian agriculture as we speak.
Have you ever been to India, Alan?
No, I haven't.
Neither I.
You'd love it. I mean, the food would draw you back. It's an absolutely wonderful experience.
Well, it sounds great. I've heard many good things from people who have visited. Someday I shall get there. But for now, here is TWiV.
Right.
Are you happy to be back with TWiV?
I am overjoyed. I miss the show. I miss being on it. I miss listening to it. I miss the whole thing. So thank you very much.
Many people have written and complimented your participation, so we're happy to have you back. We have a big program today, so let's get going. Okay. We could talk about India a long time. Maybe we'll have a TWiV just devoted to your recollections, and we'll put pictures along with it.
I will put our audience to sleep. I don't think we should do this, Vince.
Okay, we have quite a few things to talk about, and then the usual reader email and our picks of the week. So let's start with just a little recommendation for everyone. If you haven't checked out Morbidity and Mortality Weekly Report, do so. MMWR. It's a weekly publication of the CDC, the Centers for Disease Control, and it has information about specific diseases that are reported by the states and the territorial health departments, reports on infectious chronic diseases, environmental hazards, natural human-generated disasters, which we have, anything bad that happens to people that can be investigated. So it's a great resource. You go there and you can learn what's going on. And for example, in the case of viruses, you can find out what's going on in the latest outbreaks. It's not all infectious diseases, but it's really, really good. I subscribe to the email distribution. And for our listeners, I think you would love to take a look at that. We'll put a link in the show notes for that.
And a great source of conversation for cocktail parties, too.
No question.
Absolutely. Yes. Now, this past week, they published their immunization recommendations. And I thought we would just mention the viral ones. This is for the U.S., for kids, children. Hepatitis B,
Right.
polio in the form of IPV, inactivated polio vaccine.
Right.
Influenza yearly. That's a yearly
Really, for
thing.
children.
Starting at children and forever. Because we get flu every year because the strains change, right?
This is all true.
Hepatitis A, HPV, human papillomavirus.
Right.
Good one. Yeah.
It's a new one.
Yep.
Measles, mumps, rubella, which
Yep.
is one
Yep.
vaccine, and varicella, chickenpox. Those are the main kid vaccines. And then if you're an adult, you could, and you're over 50, 55, you can get a zoster vaccine, which is similar to the varicella. It's designed to prevent the reactivation of herpes that leads to zoster. So you can see those at the MMWR site. I think it's very useful. They actually have these very nice 8x10 cards that you can print out with all the vaccine recommendations. And if you're a physician, you can put it on your wall. But if there's something even better, if you're a physician, there's an application for the iPhone called Vaccines.
Oh, wow.
And this is just a cool little app. You put the patient's age in, and it tells you what vaccines they should be getting. I mean, there are many, many cool apps for this platform, but this is just an example of one related to what we're just talking about.
Yep.
Okay, that's vaccines. Second story. This is one that Alan blogged about, so we're going to let him take the lead. And Dick, you're going to love this. Larger, older deer mice spread hantavirus better than the young studs.
Okay.
What's this about, Alan?
Well, this is about a study at the University of Utah, and there's this old sort of rule of thumb, sort of general concept in epidemiology called the 20-80 rule, and the idea is that 20% of the infected individuals in a population will be responsible for 80% of the actual spread of the disease, and then it's important to note the numbers are not really what matters here. It might be 10% spread 90% or something along those lines. The issue is it's this concept that in any population, you will have individuals who are more likely to spread the disease and individuals who are less likely to spread the disease. And the minority who are the most infectious or contagious, we'll get to that later, are the ones who are going to do most of the spreading. So it's a concept that comes up a lot in a lot of different diseases, and it's not meant as an issue of blame. I mean, that comes up a lot, but to give you an example of a blameless situation, most rhinoviruses. Vince, you probably know who spreads most rhinoviruses, right?
Kids do.
Exactly.
Yeah.
I mean, that's there, and the younger they are, the more they spread them because they don't have the same social barriers as the grown-ups, and they're always putting their hands in their mouths and their hands in other places and all that sort of thing. So this was a study at University of Utah. They were looking at hantavirus, and hantavirus is the reservoir host for that is the wild deer mouse population running around in the desert. And the question was, does this rule apply? Do we have some subpopulation of deer mice that's responsible for doing most of the spreading of the disease to other deer mice? And first of all, what I found amusing about the study was the graphics that go along with it, because in order to do it, they caught a bunch of wild deer mice, checked their hantavirus status, and then marked them with fluorescent talcum powder. So there are a couple of photos on the site, and I linked one on my blog as well. You've got these fluorescent pink and green mice running around in the desert. Absolutely priceless. And somehow the hawks did not find all of them.
So, Alan, let me ask you, they took Hanta negative mice?
I think they sampled both positive and negative mice for the study, labeled, randomized them to be different colors, and released them into the population. And then
big ones and small ones as well.
Right. They picked a wide variety of ages and sizes. So they were really trying to sample as broad a spectrum of the population as they could. And then they released them. And when they did a recapture, they caught the mice again, and they looked for two things. First of all, if they were hantavirus negative before, had they caught the virus? And secondly, did they have other colors of powder on them? So if you released this mouse and it had gone off and gotten into a fight, and pink mouse versus green mouse, then each would have a little bit of the other's color, because they would have, tussled around and transferred a little bit of it, and you would figure out who was contacting who. And of course, if they'd gone and mated, you'd see different colors of powder around the genitals. So they were able to track, and they found that the larger and older mice were doing most of the fighting and mating. And those are also the mechanisms that spread hantavirus. So it turns out that populations of rodents where you have a lot of the large, older individuals are probably going to have hantavirus spread more readily through them, because those are the ones who are most responsible for spreading it. And there are a lot of issues you could raise. For example, if you skewed the population, if you eradicated the larger, older individuals, would that simply shift the spread of the disease to some other group within the population? I don't know. But it was an interesting study that once again highlighted the 2080 rule, that a small proportion of the population is responsible for the majority of the disease spread. So
the larger, older, more aggressive mice spread it because they are more likely to get in fights with other mice. Is that the bottom line?
Get in fights, and they also get lucky more often.
Now, the way it's spread to humans is when the mice pee in the houses, right? We don't know anything about that from this study, correct?
No, this study does not illuminate the transmission to humans. Although, it's worth noting, if you were to generate a higher turnover rate in the population, if you somehow had a way of selectively wiping out the older members, you might reduce transmission among the wild mice, which would be possibly part of a control program. I mean, the larger issue there is, of course, just to keep the mice and the people separate.
Mice most likely to go inside someone's house and pee are the old males, right? Judging from human behavior.
I was actually he was taking Vince serious there for a moment. Then he winked his eye at me and expected me to understand that.
So, Alan, is there any practical consequence of this result?
Not an immediate one. The practical consequence that I could most easily see would be that you might think about this if you were implementing a mouse control program, that you would want to try to skew it to kill more of the older, the larger and older mice if you were really looking at trying to control hantavirus.
I notice here that one of the authors says, we are not proposing you exterminate larger mice because they're important for the food chain.
Yes, and of course, as wildlife biologists, they're going to say that. But as virologists, I think I would vote against the mice.
Well, as you said before, it probably wouldn't matter, because if you kill all the old ones, then it shifts to something else. So.
It's possible it would shift to a different part of the population, yes.
Yeah, I thought that was really interesting, especially the technique they use to track the different mice. I don't know if that is unique or not. I'm not familiar with fluorescent tagging of animals, but it's quite nice. I like that.
Well, we actually have talked about Hanta a little bit early on in the TWiV series with regards to the ecology of how diseases spread. Remember, we talked about drought years and years of abundance, and how that affects the food supply of the mice, and then eventually how that forces the mice to start foraging for food when food supplies and reserves are getting low.
Well, you can imagine how the older mice could be more aggressive in going after the food.
It could.
Sure.
They may be the ones that get in houses.
But it's also a numbers game, because I recall that we actually had one of the people who did the investigation out at the Four Corners region come here as a graduate student back in the 80s, and he related to me that if they found a house with more than 4 mice in it, it's likely that there was a case of Hanta. But if they had less than 4, they didn't have cases of Hanta, and they explained it by a density phenomenon as well. So there's a lot of nuance in all of this. In fact, there's even some crossover information that Alan was talking about for humans with regards to the transmission of larger parasites like worms. Very, very few people in a large community, let's say in a rural community, the kinds that I would have encountered in India, you might have 1 or 2% of the population of people harboring 90% of the numbers of worms in those people, because there's a genetic predisposition for this, and then they serve, of course, as the source for the infection for everybody else.
Okay, great. I like that. Nice story. All right, moving on. Next story, viral RNA polymerase complex promotes optimal growth of 1918 virus in the lower respiratory tract of ferrets.
Continuing on the cute furry animals theme.
Now, that was the title of this article from PNAS, which was just published this week, January 13th, advanced publication. And you notice they don't even mention the name of the virus in the title of the paper, which is interesting because everyone sees 1918 and automatically say
Spanish flu.
Spanish flu. So this is a great story. As most of you probably know, there have been a couple of really big outbreaks of flu, which are called pandemics. One of them was 1918. A lot of people died from this virus, which has been called the Spanish flu. And we didn't actually have this virus in the laboratory, but a fellow named Jeff Taubenberger, a number of years ago, dug up old samples, preserved lung tissues from 3 people who had died from flu back in the early 1900s. And he recovered the entire viral sequence from those tissues. These were paraffin, formaldehyde treated sections, and he managed to get the whole sequence, and then the virus was reconstituted from the sequence using plasmids. And this, of course, uses technology that we've talked about before. That virus is really pathogenic in mice and in ferrets. Ferrets are a good model for flu. They have a nasal structure. They have turbinates that resemble human turbinates, as opposed to the mouse. And this 1918 virus not only grows in the upper tract of these ferrets, as does most strains of flu in humans, but it goes all the way down into the lower tract and multiplies there. Now, most human strains of flu, outside of this 1918 strain, are restricted to the upper tract. This one can go both upper and lower tract. So these authors wanted to know why. So they took this 1918 strain that they had recovered from DNA, and they made reassortants with a contemporary H1N1 strain, a human strain which in these animals only grows in the upper tract. They made reassortants using the DNA technology, and they looked at each virus that had, say, a number of genes from the contemporary strain and 1 or 2 or 3 genes from the 1918 strain. And what they found is that the 4 genes encoding the polymerase subunits, there are 3 P proteins and an NP protein, which constitute the viral RNA-dependent RNA polymerase. If you put those genes into the contemporary strain, it now can go down into the lower tract and replicate like gangbusters. Really neat stuff. So apparently, the idea is that the pathogenicity of this virus is due to higher replicative abilities, also because it can go down into the lower tract. And this has something to do with the RNA polymerase. So the polymerase of that 1918 virus, when put into this other contemporary strain, can make a very virulent strain. So in this paper, they looked at sites of replication, viral titers, pathology, and I think this is a very interesting finding. So what they say is that the enhanced polymerase activity could account for the increased virulence of this virus. And that's interesting because one of the proteins, the PB1 protein, was also present in the other 2 pandemic viruses of 1957 and 1968. So there were 3 pandemics so far that we know very well, 1918, 57, and 68. And one of these 4 proteins was present in all of the viruses that caused those pandemics.
Interesting.
That's very neat. Now, we don't know why these particular 4 proteins do so well in these parts of the lung. That remains to be found. But I think also it may be a good target for antivirals.
I was going to ask.
Because we have antivirals now which target different parts of flu, as we'll talk about. But this would be a different mechanism entirely. So you might want to, as the authors say, look at this as a possible target. I'm sure people have done this already.
Absolutely.
So this is actually very hot stuff because flu is in the news now. It's the season. There's been bird flu outbreaks in Hong Kong and other parts of Asia.
That's right.
Did you, when you were in India, Dick, did you hear about these bird flu outbreaks?
Yes. They were quite aware of the fact that Indonesia and Vietnam have had a number of outbreaks, and you might say that they're sort of on an orange alert in India for this. And
they're culling the chickens as a result.
They are, but I saw so many outdoor markets with tons of chickens in them.
Can't control it. Right?
I don't think you can ever control that no matter what happened.
It's interesting to have that perspective because you know, we can sit in this office and talk, but you were there, you saw them all over the place.
I did, and all the hens were inside these cages, and on the top there was always one rooster. Sort of as the guard dog for all of his harem, and little did he realize what was happening to them. They were all ending up as chicken tikka masala.
I love that stuff.
I wanted to say something more about this, too, because we had another discussion earlier about the H5N1 strain of virus, and the reason why it's not infecting people so much, and that is, I was led to believe, at least, perhaps some of our listeners can correct this with newer data, but I was led to believe it was because the wrong sugar was present on the upper respiratory tract as opposed to the lower respiratory tract, at least for the H5N1 strain. And you had to really breathe it deeply in order to get it to establish. This virus, the flu virus that's being investigated here, the Spanish flu virus, it doesn't sound like the receptor is different, but something on the inside of the virus is different.
That's correct. Well, you need to have the right hemagglutinin. I mean, it's been shown that the hemagglutinin, the viral glycoprotein that attaches the receptor of this strain, also contributed to its virulence. But these 4 proteins, more than the HA, are the main drivers of high replication.
Right.
We don't know why the avian H5N1s are not transmitting. We talked about this last week.
Right.
It's not simply a receptor issue. They're all using sialic acid, although there is a slight difference in the recognition between the human and the avian strains. It's something more complicated than that. And that's an interesting problem which we've touched on and probably will get back to many, many, many times.
Absolutely.
Next story is measles in Europe, an epidemiological assessment.
So, Vince, why is there measles in Europe? Haven't we wiped that one out? I mean, shouldn't we have done this by now?
This is a very interesting study.
Come on, could we ever wipe it out?
Sure. I mean, WHO's goal is to eliminate measles from Europe by 2010.
And WHO's other goal is to eliminate polio from the world by 2000.
Oops.
Has that happened, Alan?
Oh, sorry, never mind.
Are there animal reservoirs for measles?
Who will say no, but I don't think you could say for sure ever, right?
Okay.
The problem, Dick, as you know, is there could be a close virus which could jump.
There could.
And cause a similar disease.
But we have the vaccine. We know who gets infected. We know how to prevent it. What's the story here?
Alan, tell Dick what's the problem.
The problem with any eradication campaign, even if you've got all the usual classical factors working for you, you don't think there's a reservoir host, you've got a vaccine, the basic problem where the rubber meets the road is getting the vaccine into everybody. And I was at an HIV meeting a number of years ago where one of the speakers held up a glass of water from the podium, and he said, if curing HIV meant getting this to every person in the world once a day, we couldn't do that right now.
Right.
And his point was that logistically, particularly in developing countries, you can't transport the vaccine to the people who need it and deliver it properly to the people who need it and ensure universal coverage.
Right.
And what this paper in The Lancet shows is, even outside of the third world, you've got a problem where You're looking in Europe, which has a functioning healthcare system, unlike third world countries such as, New York. And even in this setting, you have a fundamental problem of people who don't get the vaccine for primarily because they have decided not to. So this is a whole separate level of problem. Even if you solve all the logistical problems, you have folks who believe that this vaccine causes any number of ills.
It's a backlash, isn't it?
And yes, it's backlash because we have been so successful in getting getting measles to this point. Now people who grew up not knowing what this disease does or what it can do are saying, well, who worries about measles anymore? I just won't get my kid the vaccine, which I think is criminally negligent. But a lot of the criminal justice system doesn't agree with me. So what's happening in this case is they did an epidemiological study in Europe where it's pretty much the best case scenario. And they found that you've got these outbreaks occurring, and you have certain frequency of infections still occurring. And the bottom line of the study is that it doesn't seem realistic that we can eradicate measles from Europe by 2010, which is the WHO is trying to eradicate it from the world, and
Exactly.
You can't even get it gone from Europe within the next couple of years. That's a very, very bad sign.
There was a recent posting on ProMed also. I'm back only since last Tuesday from India, but I had a chance to look at ProMed while I was out there and here too. And the estimate is that in England there's a raging epidemic right now of measles, and they claim that there's about 3 million underprotected kids out there.
Yeah.
And you're right. It's confidence in the user public. All kinds of stories about autism induced by the low levels of mercury preservatives and things of this sort. Once those rumor mills start, it's very difficult to intervene and to correct it.
Sure. Now, that's criminal that there are that many unvaccinated.
I totally agree.
In this article, they did a 2-year study, and they noted 12,000 cases in Europe of measles. The 5 highest countries, Romania, Germany, UK, Switzerland, and Italy. These are not underdeveloped countries at all.
Not. They're overdeveloped countries.
So they say the suboptimum vaccination coverage, which is they conclude is the reason, raises doubts whether we could eliminate measles by 2010.
What a shame.
The interesting thing is they don't talk at all about why there is suboptimum coverage. And all they say is we need to identify barriers for measles vaccine uptake.
Wow.
And I mean, part of the problem is people think they can make their own decisions about whether they should be immunized.
So, Vince, what's the biggest drawback of not being vaccinated and catching measles as a kid?
Subacute sclerosis Let's start again.
Right.
Subacute.
So, Vince, what's the
SSPE. It's a big one, but you can also die of primary measles, right?
Yes,
Sure.
It can be a fatal
So those
infection.
rumors are out there too, and that feeds into the rumor mill that you see that happened over here to Jones's kid, and I don't want that to happen to my kid. But it doesn't talk about the 3.5 million people that were protected that didn't get anything except their health back.
That is a great point. No one ever says how many people were protected by using this vaccine.
That's right.
Even if you accept that a few people got sick, how about the ones that were protected? And as you know,
they're in the millions. They're in the millions.
There's an old saying that vaccines would be a much easier sell if the billions of people whose lives have been saved by vaccination knew who they were.
Right.
Yeah, exactly.
Sure.
Now, Albert Sabin used to say that all the time. People would criticize him because his vaccine caused polio in some kids. And he said, well, look at all the kids that we've prevented polio in.
And without the vaccine, look at how many people get it. So
if you stop using MMR, you have huge outbreaks again.
That's right.
Mm-hmm.
And it is simply not justified. There's no reason not to use it.
That's correct.
So, there are many, many people who
So these kids in this country that don't get vaccinated get homeschooled because you're not allowed to go to school without these vaccines.
I think in the U.S. you could probably still go to school.
The health department here where we work on 168th Street and Broadway, it's had a big sign out there just before school starts every year. It says no vaccines, no school.
But in the U.S., Dick, You can have a religious or medical
I know.
reason
No, I know.
and not have to be immunized
Understood.
and go to school. So you can get around it. Right, Alan?
Yes, I believe that's correct. And the so-called medical objections that people have should really be classified as religious, because that's not a rationally based belief. It's a faith-based belief. They've decided that these vaccines are bad, and so they're not going to get them.
All right, moving to our next story. Avian flu H5N1 is gaining resistance to antivirals. How can this be? It's not infecting people. We're not using antivirals in people. How can it be? Well, let's explore it.
Please.
There have been a couple of stories about this. This is based on a paper in Infection, Genetics, and Evolution, which I did take a look at. Mainly amantadine resistance, which is one of the 3 antivirals available against influenza virus. It's possible that the use of this antiviral on chicken farms in China has contributed to this resistance. There is some resistance to Tamiflu, which is oseltamivir, one of the 2 neuraminidase inhibitors, which it's not widespread. So basically what we have here is that these strains are gaining resistance. They're not completely resistant yet. Now, the 3 antivirals, amantadine blocks the ion channel of the virus, and the Tamiflu, Relenza, Tamiflu, oseltamivir, are both neuraminidase inhibitors. These are actually designed to mimic the natural ligand of neuraminidase, which is sialic acid. Mm-hmm. The idea was if these are very close to the natural ligand, maybe we won't get resistance because then you wouldn't be able to have sialic acid binding the neuraminidase. But it's not true. We're getting resistance to both drugs. And in fact, in humans, there was an article this week in the Times which reported that basically all the H1N1 strains now circulating in the U.S. are resistant to Tamiflu.
That's right.
Now, the interesting thing about this, it's not because of drug use that has selected for resistant variants. Apparently, the amino acid change that confers resistance to Tamiflu emerged in countries where they don't use the drug. And it emerged because there was another amino acid change in the hemagglutinin, by the way. These are both in the hemagglutinin, which made the virus infect people better. So it was selected for. And then apparently this other change was needed for whatever structural reason. And it happened to confer resistance to Tamiflu. And so these strains are now circulating in the U.S., or at least these changes are in the U.S. circulating strains, and therefore we have resistance. The H3N2 strains, which are the other ones circulating, are still susceptible, but there aren't many of those this season in the U.S., so that doesn't help very much. So what the CDC is recommending that you use zanamivir or Relenza, or a combination of oseltamivir and rimantadine. Zanamivir is the other neuraminidase inhibitor. These strains are still sensitive to it, so they're saying use that instead. So what do we think will happen if people start switching to using Relenza?
Are we starting a pool on how long it'll take for Relenza-resistant strains to come out?
By the end of this season, do you think?
Yeah, I would. I'll put $1 on March.
Really?
Basically, we have 3 antivirals. Oh, here, let's back up. Hey, guys, there is a vaccine against flu. Could we take it?
Sure.
And not take antivirals? I mean, that's the problem here, that people don't get their flu shot, and then they start to get flu, and they go to their physician, they get a prescription for one of these antivirals.
Right.
And the
Well,
real
it is worth noting the vaccine is not perfect, too.
It's not perfect, right.
You want to treat breakthrough cases.
Really well worth noting. In fact, the rumor comes out every year that some university study someplace says that this year's flu vaccine won't work because it's against last year's flu virus, and this year's flu virus doesn't resemble anything.
Right.
And so most adults either opt not to get it, but if you go to Chicago O'Hare Airport, you can get a vaccine given to you on your way to your flight.
Is that right?
Yeah, they've got a flu center right there. They just, get your hamburger over here and get your flu shot over here, and you're on your way, and you're all set.
Well, here in Massachusetts, they had several drive-through flu shot clinics this year.
Flu shot. There's got to be a connection here with drive-through shooters and drive-through. Well,
yes,
Hey, I have an idea. Why don't we have Walmart give it away for free?
Hey.
Yeah.
instead of generic antibiotics, which is what they're doing now. Have you heard that, Dick?
I have not heard this.
Yeah. I think if we immunize more people, this would be less of an issue. But we have a very shallow anti-flu arsenal, antiviral.
Right? That's right.
Let's think. How does it work for HIV? HIV is not a death sentence now. If you take the antivirals, you can live your whole life. That is because you take a combination of 3, and if they get resistance, you switch to another combo. You have 15 to 20 different drugs that you can play this game with.
Yes.
And they attack different parts of the virus life cycle
That's
too.
Correct.
Right. So if we had multiple combination therapy with flu, we'd get around this issue. But playing with 1 or 2 antivirals with a virus that is replicating so extensively in people, it's just asking for resistance.
I'd say.
And yes, we will have resistance to all of these drugs. Maybe not by the end of this season, but within a year or 2, I would guess.
Yeah.
Remember, we don't have an HIV vaccine.
Nope.
So they have to take antivirals. But
Yep.
we do have a flu vaccine. So let's try and, physicians, please advise your patients to get a vaccine. Last news item is just an update. You know, we have reported that there is an outbreak of Ebola in the DRC. And there are over 30 deaths already due to this. It's apparently a new strain, Dick. And now one of the neighboring countries, Angola, is closing off more and more of its borders because it doesn't want people coming across and bringing the virus. And there's an article in Reuters, which we'll put a link to, about that. But it's interesting because it's a consequence of having a outbreak of a lethal virus infection. You want to close your borders. Sort of like that game that... Aiden talked about pandemic. Close your borders.
That's right.
Or else live in Madagascar where nobody can get to you. Dick, while you were
gone... You can keep open your Barnes & Noble, but close your borders. Is that what you're saying?
Dick, while you were away, we did an article on lemurs.
Did you?
They have a HIV-like virus in their genome.
Is that right?
First one, yeah.
Oh, my.
You'll have to catch up on your TWiV.
Wow. Oh, yes, I will.
Okay, let's get into reader email. We got some interesting ones. We're getting more and more. Thanks. We really like hearing your feedback. Brad Larson is the writer of Molecules, which is a program for the iPhone reviewed last time. And his company is called Sunset Lake Software. And he writes, I happened across your latest podcast and wanted to thank you for the kind words about Molecules. It's responses like yours that make me glad I wrote this application. He goes on to say that his brother is doing protein work, crystallography, at the University of Alabama, and that's where the inspiration for the program came from. And then he says, as far as other scientific applications for the iPhone, may I suggest solutions from my friends at Macintosh.com. It's an application that lets you easily calculate what you need to mix to produce molar solutions. Now, the cool thing about this, I checked it out. If you put in like sodium chloride, it'll tell you the molecular weight. It queries a chemical database over the internet.
This is getting too easy, Vince.
So I think this is great because, when you're in the lab, what's the molecular weight of tris?
Exactly.
So you can just get it on your iPhone. Of course, you don't want to drop your iPhone into the solution.
No,
as you're making it, but I can see that happening.
Well, I liked just doing it, the same way I kept my lab notebook with a Sharpie and a paper towel.
Alan used to come in. We'd had weekly meetings. He'd come in with a little square of a photograph, an inch square, and put it on my desk and talk for an hour about it. You remember that, Alan?
Yes.
I said, where's your notebook? He said, it's all up here.
Dear.
I did update it to, about once a month.
You actually had a computer notebook.
Yeah, I would sit down about once every couple of weeks. I'd sit down with my pile of scraps of paper and inch square photographs and transcribe them on the computer and print them out.
You were the first. When I was a student, the first week in Peter Palazzi's lab, I was a PhD student. I was putting my notes on paper towels, and he came into my office, and he saw them all. He goes, what's that? I said, it's my notes. He says, no, you will not do that. You will put them directly into your notebook because you're never going to transcribe them. And, of course, back then we were scared of our mentors. So I said, oh, my gosh. And I never did that again. And now whenever I see my students doing it, I remember those.
Well, I have a great notebook story, if we will allow just one
of course.
Anecdote. René Dubos is now given credit for being the first person on Earth to isolate and describe an antibiotic from soil. He isolated gramicidin. How do we know that he was the first and not Alexander Fleming? And we know this because he dates his notebooks. And they found the date that he did the work, and it preceded preceded Alexander Fleming by 2.5 years.
No, it's really important.
So there it is.
You have to date your
It becomes
notebooks.
a legal document after
Absolutely.
a while.
No, notebooks are accepted as legal documents for sure. You got to date them. Of course, technically they should be signed at the end of each day by a witness, but
we all do that.
yeah, sure. Right,
or we should all do that.
So the next email is from Samuel, who's a PhD student from England.
Uh-huh.
And Samuel writes, Dear Vincent Dick, I would just like to commend you both on what has to be one of the most interesting podcasts in existence. Your show is witty, clever, and very, very interesting, full of intriguing topics. I have a massive interest in infectious disease, and in particular those caused by viruses, so this show is right up my alley. It keeps me going on the long train journeys to and from university.
Outstanding.
This is what we love, don't we?
I tell you, this is good feedback. Of course, we're not going to read you all the other letters we got that were very negative. No, that's not true. Actually,
and of course, he doesn't mention my name. So,
well, he probably was an early episode.
That's true.
I would like to put forward a suggestion for a future show, perhaps something on our great achievements in the field of virology. I'm thinking particularly about those viruses we have been able to largely eradicate, and how we have achieved this, e.g. smallpox, polio. Perhaps a focus on vaccines, what they are, how they are made, etc. Keep up the good work. It's a great idea.
Yep.
We'll do that. And if you listeners tell us what you think are the great achievements in virology of all time.
Yeah.
And I do think smallpox eradication is, of course, one of them.
Oh, absolutely.
There are others as well. Everett writes, Dr. R., the first link is an article which you have seen, and that is, in fact, he sent us the link to the article on 1918 flu, which we just discussed above. So thanks for that link, Everett. And he sent us something else which will come up in a few moments. And now he has a question. My question for the week relates to the strandedness of viruses. Do viruses that are, say, negative single-strand RNA more fit from an evolutionary standpoint than ones that are plus single-stranded RNA? Same for single-stranded DNA versus double-stranded. Is there any advantage for the virus to be one or the other? And then he says, if fitness is defined in terms of adaptability, wouldn't flu be amongst the most evolved virus due to its mutability? It's a great question. You could talk about it for a whole show. I don't know of any advantage to being negative or positive strand. Certainly it's better to be RNA versus DNA because the error rate is higher for RNA polymerases than for DNA polymerases.
And if you're a virus, then errors are good.
Errors are always good because that drives evolution. DNA, interestingly, there seems to be a slightly higher error rate for single-stranded DNA viruses than double-stranded DNA viruses, and I don't know what the basis for that is. So if you had to choose, you should be an RNA virus or a single-stranded DNA virus. Now, the question about flu, the most evolved virus, well, certainly flu can evolve. Just like any other RNA virus, it makes a lot of mistakes. The neat thing about flu is that apparently its glycoproteins can accommodate enormous changes. Which lead to us having to make a new vaccine every year, for example.
Right.
Not all viruses can do this. Like measles has glycoproteins as well. It's an RNA virus, but it doesn't undergo the same extensive change in its protein sequence. I don't really know the reason for that constraint. So there are major differences. Just being error-prone is not enough. There are some other issues that a virus has to overcome.
What about speed of replication? Do RNA-stranded viruses replicate faster because they get a jump start on the machinery, or do the DNA viruses eventually catch up in speed?
Well, there's a range. There are some DNA viruses that are pretty quick. They can match RNA viruses in terms of speed. Many of them take longer because their genomes are longer and they have to get into the nucleus and so forth. Yes. So in general, the RNA viruses are faster, and they can have more generations and therefore evolve faster.
It's also worth noting, I mean, the question implies in asking if influenza is among the most evolved viruses, there's not really a hierarchy.
Right.
What is the most promiscuous virus you can think of?
What do you mean by promiscuous?
With the widest host range. See, that's the way I would define highly evolved, because if you can take advantage of any life form, and I think we talked about West Nile for a while, in fact a lot. It infects cold-blooded invertebrates, warm-blooded vertebrates, and look across the range of host species. There isn't a single animal out there that isn't susceptible. How more evolved do you need to be?
Well, it's a good way to look at it. Some people would say evolved as occupying a very specific niche also. So I don't know if infecting
That's
a lot of
too
hosts.
highly evolved.
You think so?
Well, then you risk extinction when your host.
You could also define evolution in terms of complexity, in which case the pox viruses and the memeviruses would come out at the top.
Mm-hmm.
Gotcha.
Well, I wouldn't say flu is the most evolved because personally I think they're always evolving and they haven't reached an endpoint. There's no endpoint to evolution, right? It's just a continuum. So flu has a great capacity to evolve because it makes a lot of mistakes and it makes a lot of particles and it reassorts. Those are the drivers of evolution. In terms of your question, Dick, I don't know what virus would infect the most number of species. I don't want to guess, so let's look at it and get back next week.
Maybe one of our listeners knows.
Tell us.
Yes.
Absolutely.
The top 10 promiscuous viruses are. We can give this to David Letterman.
You want it to be according to host range, right?
Yeah.
Okay.
Yeah. Absolutely.
I know the papillomas infect most mammals, most vertebrates.
Do we know any plant viruses that also infect animals?
At the moment, no, but there are some hypotheses of jumps that have occurred.
I remember meeting Peyton Rouse when I was at Rockefeller. He was in his last year there. And the tobacco mosaic virus and the Rouse sarcoma virus, all those old filterable agents that have, gained fame by just simply being discovered before all the others. The easier ones came first, I presume.
Yeah. Well, you can still find them, though, Dick, as you know.
No question.
All right, a couple more emails. Stu is a medicinal chemist. I'm a synthetic medicinal chemist who, for a number of years, has been involved with antiviral drug design and discovery. I just wanted you to know that your TWiV is excellent, very informative, and engaging for this organic chemist. Your efforts are building bridges to other fields. That's great. My research group of PhD students and postdocs, organic chemists, are also required to TWiV via iTunes. Thanks, Stu.
Awesome.
That's great.
You have our deepest sympathies.
Stu is the students, right, and postdocs?
That's right.
Stu is at Auburn University.
Nice.
Chad writes, my name is Chad, and I recently graduated from UC Davis with a BS in neurology, neurobiology, physiology, and behavior. Unfortunately, I figured out I was into micro, mainly virology, too late into my studies to change major to micro. I did, however, take as many virology courses as I could at Davis and other various courses dealing with microbial pathogenesis. And he used my textbook.
Outstanding. You have a new edition, by the way, I understand, Vince.
It is.
Congratulations.
Thank you.
Congratulations.
I am fascinated by virology. While I am glad to be done with my undergraduate work, I would like to apply for a PhD in virology one day. So basically, he's asking for recommendations about good virology programs. And no, we haven't covered that, but we will. We'll think about it and come back to you with some suggestions for good virology programs. And the last email is from Patty, who writes, love the show so much that I've begun an introductory medical virology course as part of my continuing education.
Nice.
Obviously, I'm not quite in the caliber of a lot of your listeners, but your podcast has made me curious, and I enjoy the banter and also the way you present the scientific discussions. Yeah, we bantered today, that's for sure. Can you perhaps elucidate a minor point? I'm having a bit of a problem getting good definitions for contagious and infectious. And he's looked on the web and he finds that they're interchangeably used. So I answered him, but Dick, I'd like to know what your definition of contagious and infectious would be.
I use them
interchangeably.
They're synonymous. I think the difference is that they're spelled differently.
Okay, that's a simple answer.
But there's a bigger confusion out there, especially with beginning students in infectious diseases. What's the difference between an infection and a disease? And
the big difference, right?
You start out with an infection, and sometimes you end up with a disease, but
you may not.
You may not. That's right. And so people do tend to confuse those two terms in the beginning. But,
okay. Dick, Alan, what about you? What do you think?
I use them. I see your answer in the notes coming up. I use them yet a different way, which is I would refer to a host with a virus as being contagious, or it's a contagious disease, whereas infectious I would use to describe either the virus itself, it's an infectious virus, or any substance you might be dealing with. So you would say you have infectious waste coming out of the hospital. It's not contagious waste, it's infectious waste. But again, I think it's a little nebulous.
Sure. No, they are overlapping.
Yeah.
I tend to use contagious to describe a person who's infected. They're contagious. I wouldn't say a virus is contagious. That's not correct,
Mm-hmm.
although some people might use it. An infection, as you do, Alan, I reserve for the organism itself. Poliovirus is
right.
infectious.
Sure.
I know we use infectious waste, but I don't particularly like it. I would say it's contaminated or something like that because it's the virus in the waste that's infectious. But usage is that, yes, you do call waste infectious.
Yeah,
Right. And contaminated waste doesn't specify that it's biologically contaminated.
Exactly. So they're both adjectives. Contagious refers to the host and infectious to the organism or something contaminated with it.
Or when someone's in a recovery period from a viral infection, you can say they're not contagious anymore, or they're not infectious anymore.
That's correct.
So, the physicians are prone to using these terms more loosely, I guess, than the virologists.
Yeah, the physician would. You're right. They would interchangeably use contagious and infectious.
You're no longer contagious. You're no longer infectious, but you've still got the infection.
I mean, it's not incorrect,
right?
But the way I would do it is
I think your way is more precise than it is.
All right. That brings us to our picks of the week, and we'll get through these quickly because we have held your ear for a while.
We've wasted nearly an hour of your time.
Yeah, they're learning, Alan. They're learning.
Oh, yes.
Our science blog of the week is a site called Molecule of the Day. And this site is by a chemist who enjoys rambling about the relationship between chemistry and real life. So, for example, in a recent post, he talked about loratadine, which is an antihistamine used for treating allergies. So I like it. Every day a different molecule. Maybe not always molecules, but you get the picture. Science podcast of the week. Meet the Scientist with Dr. Mary Buckley. This is an ASM-produced podcast. She interviews a different scientist on every episode, and if you like hearing scientists talk about their work, it's a good podcast. It's well done, it's clear, and she talks with interesting people.
Great.
And finally, Science Book of the Week. This was suggested by Everett. Thanks, Everett. The Great Influenza by John Barry.
Good choice.
The epic story of the deadliest plague in history. We've talked a lot about the 1918 plague, and this book is a great description of it. Have you read this, Dick?
I know the book. I read parts of it. I haven't read the whole book.
How about you, Alan?
I have read it. It's a very, very well written.
My former chairman gave this to me a few years ago, and I read it. It's very, very good. Highly recommend it.
Speaking of books,
yeah.
What is this the 200th anniversary of? Publication of Origin of Species.
Origin of Species?
Yeah,
Origin of Species, yes.
This month or
doesn't seem
year?
Possible, does it?
That was this year.
That's this year.
2009. Yeah. We'll put a link in the show notes to buying a copy at the TWiV bookstore.
Great.
And I believe the author's 250th birthday is coming up.
Indeed.
Would that be Chuck?
Yes, that would be Chuck.
All right. I am off to Switzerland on Wednesday, and
Sounds
I am
good.
going to talk with 3 virologists and record 3 TWiVs. So we won't TWiV on Friday, but next week we shall. And those will be interesting virologists. You can reach me on Twitter at PROFVRR. Alan is Alan Dove on Twitter. You can hear us twittering about science mainly. It's been a very good medium for finding out new things in science.
Yeah.
Tell us what you like to hear about on the show. TWiV at twiv.tv. That's our email. We have a website, www.twiv.tv, where you can download all the episodes and read the show notes. You have been listening to This Week in Virology. Welcome back, Dick.
Thanks, Vince.
Nice to have you back. Thanks for joining us, Alan.
Sure.
See you next time. Good night.
Good night and good luck.